questions?
You might confuse.
Is that working?
Yeah.
I'm just, when nanotechnology was first spoken about, the element mechanical engineering, the promises it was making with regards to materials, with properties like its stiffness, ridiculous factors over and above the strongest kind of materials you have today, like titanium or even composite materials. It was up to about, I can't remember the factors, but they were 20 times as strong. You're kind of saying, well, you're going to have airplanes that are, and skyscrapers that are, possibly reduced in weight by 50% or whatever like that. What has prevented that amount of scaling? Is it being able to, the inability to be able to process the materials in a commercial fashion, or is it the safety aspects that have not been addressed? What's holding that back?
In terms of composite materials and nano-composite materials, certainly when nanotechnology first came, it promised the sun, moon and stars, but all technologies do that. That is a catch-22 for scientists, that in order to get funding, you need to promise, and not only in terms of get funding, but also to get a scientific discipline established, you need to promise it's going to have to change the world in some senses. So to be honest, I would say the composite materials and nano-composite materials is one of the areas that is actually working relatively well, and there are actually applications out there. They're just not hyped for the simple reason that they are what is called enabling technologies. It's not a new product. It's just a slight change to an existing product. So there are already cars out there that are using nano-composite materials. There are airplanes out there using them. But because for the consumer, the person buying it, it doesn't matter. All they want is a product, a car that drives. If it's cheaper, great. If it's scratch resistant, even better. If the color doesn't fade because there's pigment in the nanoparticles, wonderful. They don't care how it does it, in general. So I think in cases where the nano aspect of it is to enhance an already existing property, they are out there. They are on the market. Now, I don't know, I couldn't tell you off the top of my head whether this factor of 20 in terms of stiffness or strength has been achieved. But in terms of products reaching the market, they are out there. But there's a difference between a nano-enabled product and a nano product, I would say. And I'd say that's probably the best answer I can give you at the moment. So in that case, there is still a safety element, but it's much more, it's what's called the product life cycle. So it's more at the end of the life of that product. So for example, when the car gets scrapped, is there a potential for those nanoparticles that were added to it to come out as the material degrades over several hundred years? But that's not an immediate safety concern. So that's for what we call embedded nanoparticles, that's not a factor in delaying their products getting to market. So, okay?
Anybody else?
Hi, you say you can design these nanoparticles to bind, say, particular chemicals or to bind specific locations in the body. If something binds with something else, does it have to have a particular shape? And how do you design the nanoparticles to have this particular shape or to bind? And the second question, I guess, really is, in your day-to-day job, do you go in and do you spend a lot of time in a lab, like with a microscope, and playing around, or do you do a lot of computer simulations, or maybe some comment on that?
Perfect. Okay, so the first question was how do we design particles to do specific things? So because I said nanoparticles have an enormous surface area, this means that they just bind in what we call a non-specific manner. So they will just bind things, because the same way that they say nature abhors a vacuum, it also does not like a clean surface. So there's no such thing as a clean surface. You know yourself when you wipe, 2 seconds later, the dust is already starting to stick again. So when I take a nanoparticle that I've made, and I've either made it in some specific solvent, or I've made it in water, or I've made it however I've made it, as soon as I take that then to the next solution, so say for example I want to introduce it into a cell, that means then I have to disperse it in the liquid or the broth that my cells grow in. And that contains proteins, it contains salts, it contains all sorts of things. So immediately I put my nanoparticle into that solution, it will bind stuff. It will bind proteins, it will bind salts, it will bind, if there's a colored dye in my solution so that I know that my solution is fresh, it'll combine that too. So the trick, as you say then, is to design the surface such that it binds the things you want it to bind and not the things that you don't want it to bind. And because there's what's called an affinity. So if I have an affinity for you, I want to stand beside you. Whereas if I have no affinity for this poor man over here, no, I won't necessarily. So, but what happens is I might meet you first. So I'll have an interaction with you first. But then I'll have a stronger affinity for this person. So after a while, I'll drift this way. So it's the same way, that things bind initially, possibly because they're abundant, there's a lot of them, or just because they happen to touch each other. But then there's things that have an affinity, and they will gradually replace the things that bound first. So that's what's called, it'll reach an equilibrium. So eventually it'll find the things that it wants, and it'll discard the things it doesn't want. But then we can take it a step further, where we can actually say, okay, I know, for example, that there's a whole gradient of attractions based on whether things are water-loving or oil-loving, for example. So I can design, if I know, for example, I want to bind fat, for example, like cholesterol or something like that, I'm going to make a surface that has similar properties, so that it's going to be oil-loving or fat-loving. So then that will attract. So there's a whole range of known chemical type interactions that I can then program into my particle surface. So I can use, I mentioned already, the positive and negatives attract. So I can, for example, put positive charges to attract a negative species, or the other way around. Or I can even play with my surface such that I have some positives and some negatives, so that I can even then start to direct it to the certain part of the surface that I want it to go to. And you mentioned shape recognition. So biology works, like your cells and protein interactions and things often work on these very specific shape recognitions. Like this lock and key, it's called. One key fits one lock. So I might have 1,000 keys on my keyring, but I can only open my door with one of them. And this is used in biology an awful lot. And there are some few examples in chemistry where we can mimic that, but they are very few at present. We are not nearly as good at it as nature is. But then nature has had billions of years to evolve these specific. So there's a classic example would be, say, like biotin and streptavidin. That's a really well-known pair. They just, if you have a surface with one and the other is present, it'll pull it out of solution with extraordinary affinity and capacity. So it'll bind that and nothing else, basically. So, but we are still quite a long way away from knowing all the rules to design these particles, but we're getting there. We're getting there. So then the other question was, let me recap.
What do you do day-to-day?
My day-to-day life. Well, my day-to-day life for me is less in the lab than I would like it to be these days. But I manage a team of people who are in the lab on a daily basis. And our research ranges all the way from making the nanoparticles, so actually in the lab doing chemical reactions, through to characterizing the particles, which is basically looking at them under a microscope, which is looking at how they interact with proteins. Then the next step in we sort of have what we call a research loop that everything that we make goes through. So the next step in the research loop then is looking to actually visualize where the particles go if we introduce them to different cell types. So looking at how they get taken up, how quickly they get taken up by cells, where they go in the cell, And so that involves then that we have to design particles not only to be functional, but also to have a tag or a label that will report back to us. So one example is using a fluorescent label, which means that we shine a light, a laser of a certain wavelength on the particle, it absorbs the energy, and then it emits that back out as a color that we can see. So that's another level of complexity then that we have to build into our particles so that we can track them when they're interacting. Then we want to know, so then after we've discovered where in the cell our particles go, we want to see what they do there. So then we look at things like, you know, are we getting changes in how much of a certain protein is present? Are we changing how the cells signal to one another? Are we making the cells happier, for example? Do the cells divide better? Or are we making them slightly less happy? Or are we not having any effect? There are materials that go in and have no effect. The cell doesn't even know they're there. So those are the great ones. So unless, of course, you're looking for a specific effect, which would be, for example, that you want to target and kill a cancer cell, then you want to be able to do that. You want the particle to trigger some reaction that tells this cancerous cell to shut down. Then we also do, so that's sort of our standard loop. And then, for example, we'll say, okay, instead of just looking at a cell, I want to look at, say, for example, a simple aquatic organism, so like a little water flea. We want to say, okay, if my nanoparticles are released into the environment, do they interact with the water flea? Do they interact with algae? All the way through then, do they interact with larger animals? Do they interact with soil? So we have groups doing sort of what we would consider the whole loop of things we want to know. And then it's also like it goes back to the start, because then we go, well, okay, the particle that we thought would do this wonderful thing and go this wonderful place didn't quite get there, or it didn't get there with 100% efficiency. So then we'll go back and maybe redesign our particle a bit more, see if we can change some of the proteins it binds, because it's our belief is that it's the proteins that signal to the cells what to do with the particles. So but my day-to-day life is telling the students what to do more than actually doing it myself at this point, I have to say. So.
Iseult, I suppose I was just wondering, do you have any feeling as to what the public perception of nano is? Do they realise that it's out there? Are they happy, sad about it? And I suppose, is there anything from the genetically modified organism experience, I suppose, that you think maybe nano can learn from?
Sure. That's actually a really good question, and I think that's why I emphasize to some extent that nanotechnology is actually possibly the first technology where public perception and also managing the safety aspect is being developed in parallel with applications. And this is mostly because of the experiences with genetically modified organisms, where there a lot of the high-tech biotech companies forged ahead going, this is the way we're going to do it, and they didn't necessarily wait to see if public perception was sufficient, if the public would accept these sorts of genetically modified crops. So, which it turned out in Europe we don't, so, or we haven't as yet. So there are studies on how aware people are of nanotechnology. There hasn't been any in Ireland yet, and it's something I would be very interested to do. But there was a big study by Which magazine, you know, the Consumer Choice magazine in the UK. There they basically formed focus groups, and they asked initially each of the people coming in how aware they were of nanotechnology, how positive they felt towards nanotechnology. And then they gave some examples. So they said, what about nanotechnology in food packaging, for example, or nanotechnology directly in your food, nanoparticles in your food. And then they mentioned, of course, medicine and other examples. So interestingly, people, probably for the same reason they were a bit queasy about genetically modified organisms, people are a little, at least in the UK, people were a little bit less receptive to the idea of nanotechnology in food. Everybody in that study was very positive about the potential benefits of nanotechnology for medicine and for telecommunications and for I believe they had environment in that as well, but I would have to check. So the one that came across as we'd like more information at least, or we'd like the same, we'd like it on the label, is food. So, but overall, I think people are positive about nanotechnology. But I think the complication is, again, that there is a significant difference between a product that's nano-enabled, which means that somewhere in the process there was nanotechnology, versus an actual product that this product has nanoparticles. So an example of a consumer product that has nanoparticles is sunscreen, for example. Some of the cosmetics where they talk about liposomes and improved dermal delivery, and a lot of that uses nanotechnology. But if you consider, in the case of sunscreen, for example, the statistics are that if they took the nanoparticles out of sunscreen, the rate of skin cancer death would be so much higher than it is, so that even if there is a slight unknown as to the long-term consequences, they still outweigh the short-term removing it. And my feeling on all of this is, I use sunscreen. Well, I don't, because I'm pretty bad, but when I'm on holidays, I do. So, and I don't know of any example where there is an actual safety risk at the moment. All I know is that there is potentially some risks involved with some nanoparticles in some cases. So overall, I think public perception is good, and hopefully it will stay that way if scientists, I think, continue to engage with the public and encourage the public to understand the benefits. I also think myself, in terms of things like, well, Botox, for example. I haven't used it and I never would, but I know of lots of people who make the personal choice that they will inject botulism into their forehead to prevent aging. So if they're willing to do that, I don't see why you wouldn't be willing to use a cosmetic that has nanotechnology in it. But that's my personal opinion, I guess. So, will I jump or will you jump? Okay.
What attracted you to this field of research in the first place? And perhaps tell us something about the project that you're working currently on.
Okay. Well, about 2 years ago, the group that I work in won a prize in the US, and they asked me the very same question: how did I get involved in nanotechnology? And I'll tell you the answer I gave there, even though it wasn't a politically correct answer. I will say the answer is that we started off working in biomaterials, and as we were trying to develop materials to have specific functions, the materials were getting smaller and smaller and smaller. And 5 years ago, we weren't really using the word nanotechnology. We were doing more or less the same thing, but the particles were maybe slightly bigger, so we were calling it microparticles instead of nanoparticles. So basically, I just drifted into it as my particles got smaller and smaller. So, so that's pretty much it. I was working, for example, on my PhD, I was working on polymer coatings for drug delivery to coat, for example, stents that go into your heart during a heart operation, or to re-expand your arteries after they've been clogged. There we were basically looking to control how fast or how slowly the drugs were released into the bloodstream. And eventually we got the idea that many of these processes are sort of it's just diffusion, how quickly the molecule just moves on its random path through the polymer matrix. So then we thought, well, what about if we make a really dense polymer matrix inside the other polymer? So then we were making sort of particles inside our polymer. And then we discovered that if we make the particles smaller and smaller and smaller, we can make them denser and denser, and we can control in that way how they came out. So I sort of drifted into it. Then the other part was, a current project. At the moment, a lot of our research is focused on the safety end, for the simple reason that it's a more short-term issue, and potentially applications in nanomedicine and IT, unless we can resolve the issue, because at the moment there's a lot of uncertainty. And uncertainty is bad for business. It's bad for scientists. It's bad for everybody to some extent, because an uncertainty is essentially the same thing as a risk. So at the moment, most of what we're doing is directed towards answering some of these questions that can we predict where nanoparticles will go? So as I said, there's a range of different materials that they can be made from, a range of different shapes. Then we can change the surface slightly, then we can functionalize them to have another surface group, then we can add labels and so on. So they say there's something like 30,000 nanoparticles in various labs around the world. And it's not really feasible to test each of them in the way that we would test chemicals at the moment, where there's a whole series of safety evaluations. So a lot of what we're trying to focus on now is looking for patterns. So if I have a certain surface charge, this causes certain proteins to bind and results in my particles going to a certain place. So then possibly the underlying material doesn't matter, it's what I've done to the surface. And if we could then develop sort of screening tools so that we can look at a whole bunch of materials, the same way that, say, for example, the pharmaceutical industry looks for potentially new medicines. They make hundreds of chemicals, hundreds of drug molecules, all just slightly different, and they screen them rapidly to see which one is the best candidate to treat that. So a lot of our research is focused towards developing these sorts of screening tools for nanoparticles and developing then predictive software or predictive approaches to tell us, okay, this nanoparticle is suitable for nanomedicine, this one is less suitable, this one is suitable for application in IT, and we can coat it in this material to make sure that it doesn't ever reach, a cell or something that it shouldn't reach. So but of course we are always, once this safety issue is resolved, which we believe it will have to be in the next couple of years, then our longer term goal is, okay, what are the applications? And what are the applications where we as a small group in a small country can make a difference rather than constantly being behind countries like Germany or the US, where they have more scientists. I don't know about more money at the moment, but certainly more scientists working on a topic. So we're always looking around going, what can we do that's different? What can we do that keeps us ahead of the game? So that's, yeah.
I would like to bring up an issue, since it wasn't specifically mentioned yet, which is regulation. As you know, if there is a potential nano hazard, authorities are obliged to come up with rules that protect people from being exposed to products that might come up in the industry without regulations. So what's in the pipeline there that you have in common?
Okay. So at the moment, because nanomaterials are generally made from existing chemicals, they are considered as chemicals. So the only difference typically is that we've made them smaller. So they are regulated currently as chemicals, which means that they have to go through a registration process, they get evaluated, and they get approved, I guess, eventually. So that's, for Ireland, that's generally done at a European level through what's called REACH. This is a directive from Europe. So there is, as I mentioned, there are some properties about nanomaterials that make them slightly different. So one of them being their large surface area, one of them being their access to new places, and one of them being their access, their surface, and their reactivity, so that they can be more reactive than a larger equivalent. So currently there are many groups, including ours, working on just applying the existing regulation and working through for specific examples of nanoparticles and seeing, do the existing regulations capture all of these aspects? So at the moment the consensus is that yes, existing regulations for chemicals cover most of it. Then if you have an application, for example, in food packaging, where you're putting nanoparticles in, for example, to trap moisture, for example, to protect your food for longer, or there's also ones where they're putting in a little sensor that will tell you if it's been exposed to bacteria, for example, or if the temperature has come up and down on a product that's meant to stay cold. So there's all sorts of neat tricks that they're doing in food packaging as well that I didn't mention. But anything regarding food then goes through a whole separate regulation. Then you have to go through all of the food regulation. It'll be the same in all of the environmental issues. There's environmental regulation that you have to make sure you're not harming the environment. And again, in most cases, nanoparticles will be treated as chemicals and will fall under the existing regulation. And if and as potential new risks related to nanotechnology emerge, they will be regulated accordingly. So, but at the moment, they are regulated as chemicals under each of the existing application areas. So, and the other one I mentioned is applications for nanomedicine. They will go through all the existing regulations for medicines, drugs, medical devices. What else could there be? Those are the main ones. So they're governed through, for example, the European Medicines Agency. So there's plenty of regulation in place. So.
Just allow me a comment on your answer there. It relates slightly to the question I want to ask. When you're talking about the benefits or potential applications, beneficial applications of nanotechnology, the great stress is on the difference of nanoparticles. In terms of the regulation, you've actually stressed the continuity. You've said actually it's just really chemicals under another name. And one would have thought that if you were to follow the logic of the promise of nanotechnology based on this difference, you would have said yes, and we will need a whole new schema for regulation of nanoparticles. But allow me just to ask a question that has to do with this idea of promise that you touched on. And it actually relates even to GM organisms, which I mentioned a moment ago. 10 years ago, we were at the height of the debate about GM organisms in Ireland. And one of the things that was being said by way of kind of conclusive argument as to why genetically modified foods, crops were a good thing was because they would feed the hungry. And actually the vast majority of the effort in growing GM crops, of course, is not in the areas where people are most hungry. You made the point about the application of nanotechnology to filtering salt out of water. And you made the perhaps mildly demagogic point that there's over 1 billion people who don't have access to clean water. And yet when you mentioned the areas where this technology was actually being used, it was Australia, Florida, and California, and they're not the regions where the 1 billion plus who don't have access to clean water are concentrated. So are people in the nanoscience community concerned about the socially responsible application of this potentially very promising technology to the greatest benefit of mankind?
That is a really, really good question, and it's something that, again, most technologies suffer from, that they have the potential to widen the gap between those that have and have not. There's something called the Foresight Initiative, the Nanotechnology Foresight Initiative, which about 5 or 6 years ago came up with 10 grand challenges for nanotechnology. And many of them were related to social equality and to using the technologies for the benefit of more than just the developed countries. So when I mentioned the desalination, those existing ones tend to be large-scale plants, where it's actually like an entire huge compound that's doing it. The real promise of nanotechnology, and where the real benefit for somebody in a rural community that doesn't have access to these things, is that people are working on, and they're still at lab scale, people are working on devices that can just be used in a regular plastic bottle, you know what I mean, or something that can be For example, I mentioned that the nanoscale titanium is triggered by sunlight. So people are working on applications where you won't need a power plant to run it. You won't need a power plant to drive it, that it will be able to, and that's again where it's this combination of capturing natural resources and using them then. So in order for it to not require a large power plant, we're still some way away, but the dream is there and the challenges are there. So that the purification of water is one of the 10 grand challenges for nanotechnology. Another one relates to more efficient use of soil and land in general to increase production yields. And for example, applications in agriculture are quite promising for the same reason that we can, say, control delivery of medicines. The promise is that we can control delivery of fertilizers or insecticides or any of those things which are bad for the environment. That again, if we can use nanotechnology to deliver these in more controlled doses or over time so that we don't have, you know, large scale spraying. And again, those sorts of applications are being developed with less rich soil places in mind or with areas where soil is damaged. And again, the remediation one is of interest to me personally, because when I was quite young, and the reason I mentioned war zones was because when I was quite young, my father used to work until he retired for the Department of Pesticide Control in the Department of Agriculture. And when I was quite young, I remember him going to Vietnam, and he was brought in as a consultant to work on a project to see how they were going to remediate the soil that had been, I think it was napalmed or not napalm, is that what? So which had been blanket sprayed with nasty, nasty chemicals. And the soil, if they just left it as it was, I think they were saying it was going to take something like 200 years for the soil to be usable again. So for me, that's one of the personal interests in, you know, environmental applications. So, but yeah, I believe that in the longer term, the technologies will benefit the wider communities and hopefully narrow the gap between those that have and have not. So.
If I ask you a question about ourselves, therapeutic, say for example viruses, you raised the spectre, if you want, earlier, that you could, for example, have nanotechnology, it could be tried out on us so we could find that it's a problem. But recently there was a movie with Will Smith last year in which most human beings are returned to a feral type of state whereby their aggressive tendencies are, if you want, accentuated by something, not necessarily, I hope not, nanotechnology. But I'm wondering what is known, and empirically, how do you test these things? I mean, you have to really test them on mice first, don't you, then primates, then ourselves, and then linked to that, probably the most virulent pathogens are in some ways viruses, aren't they? So how do nanos, so to speak, interact with viruses? Thank you.
Well, I'm sure most of you have heard of the grey goo phenomena, where there was, I think it was Prince Charles that mentioned it first. So the main difference between viruses and nanoparticles is that viruses replicate. They are designed, they go in, they spin out a whole bunch more of themselves, and therefore they take over, and that's how they work. Nanoparticles are several lifetimes away, if ever, which I actually don't believe that materials will ever be able to replicate themselves. So, because that would go against the basic laws of physics, that you can't make matter from nothing, essentially. So, one of the things that we're doing in our group is utilizing or studying how viruses work. Because viruses, what they have is they have a surface of proteins, and the way that they evade your immune system is that they're constantly changing this surface. So just as your blood cells, the white blood cells, or the cells that are designed to capture these, once they have figured out the pattern on the surface, this lock and key thing to pull in on them, the virus then will mutate or change its surface so that it's slightly different. And that's, of course, the risk that people are worried about with this swine flu, that it'll mutate slightly, and then the vaccines that we've developed to it won't quite fit. So if we can understand what the range of targeting molecules that viruses can produce, because it's hard to believe they can produce any combination. It's possible they can, but it's hard to believe that they would be able to produce any combination. So likely they have a panel that they can build. So if we can figure out what these are and develop synthetic ones so that we can target our particles using those same pathways, that's the ultimate goal. But the key point is that once we have put a specific label or a specific tag on, that's not going to change. Our particle isn't going to be able to then, if the cells respond in such a way that they say, okay, we don't want this particle coming in anymore, so we're not going to make that receptor anymore, then a virus can adapt, but a nanoparticle will never be able to adapt. So while we can learn from viruses, we won't be able to have that evolutionary part in it. Does that answer your question, or did I waffle?
What you didn't know, the second part is how do we test all this? We're not going to use ourselves as guinea pigs, really, are we?
No. And in fact, there's directives to say that we should even minimize the amount of animal studies we do. So again, I mentioned that a lot of what we're doing is looking to see if we can develop screening platforms. And they would all be what we call in vitro. So they're just like on plated cells. So obviously, even for existing chemicals and for pesticides and drugs, we are not at the stage yet where we can remove animal testing. So even for classical compounds, there is still a need for some animal testing. And some of the regulatory processes, some of the standard tests you have to do are, for example, you have to do a skin test on, I think it's rabbits, but I'm not sure. There's also what's called biodistribution, which is where you, say, for example, inject the chemical you're looking at into the tail of a mice, and then you're looking to see where in each organ where it goes, how long it takes for it to come out, for example, through the urine or through the feces. You're looking at which organs it goes to, whether it goes to the liver for processing or whether it goes to the kidney. So generally, we will be testing nanoparticles using the very same tests. And this comes back to the regulation, that at the moment we will use the very same tests and additional ones as necessary. And eventually, we hope, for at least in the early screening stages, to be able to do a lot of it just on cells. So. Again, this is where I say you can't, there's no such thing as a nanoparticle that's representative of all. So some, depending on what it's made from, some nanoparticles dissolve. So for example, a nanoparticle made of silver, eventually it will dissolve, you'll just get ionic silver again. So that particle will disappear, essentially, it'll dissolve. Many polymeric ones that are designed for nanomedicine, for example, are designed to degrade, so you'll end up with the polymer unparticleized. It'll sort of just go back and it'll disperse. So yes, those sorts will degrade. Things like, like a gold nanoparticle, that is at least not in the short term going to degrade. So more likely it will be cleared through the kidneys, or cleared and excreted rather than degrade in that sense. So because these tend to be like polystyrene, like your polystyrene cup. I mean, one of the problems we have with these engineered materials of all sizes and shapes is that they degrade over very, very long times. So this is why we end up with landfills and all sorts of things. So again, these problems are not new problems, and they're not specific to nano, but they do exist for nano. So does that answer that? Yeah. Okay.
Okay, let's take this opportunity to thank Iseult for coming along. A very interesting solution talk. And he always comes out to film, so thank you, Mark. And thank you all for coming on such a beautiful summer's evening. We'll keep you posted on the next cafe. I can't remember when it is. It's maybe a few weeks, we're not really sure yet. Oh, there you go, the 9th of September. So keep it.
It's Science
In
Gallery.
the Science Gallery, the 9th of September. Is that Wednesday? Yeah. Wednesday, the 9th of September, Science Gallery. Okay, so thanks very much for coming along and catch you the next time.
Thank you.